Good afternoon, I'm Sanya. I'm a PhD student at the University of Pittsburgh, and my talk today is titled Evaluation of Named Entity Recognition Systems for Ontology Concept Annotation in Biomedical Knowledge Graphs. In the next 15 minutes, I'll go over some background of ontology concept annotation, biomedical knowledge graphs, our research hypothesis, results, as well as some limitations and conclusion. Concept annotation is a subset of named entity recognition, which is more familiar to people. It is the task of identifying biomedical concepts or concepts in biomedical text, specifically such as chemical compounds, genes, proteins, or viruses, disorders. Named entity recognition is the broader category, as it can be used for identifying entities, relations, attributes, or even events. And named entity recognition tools are often used for concept annotation and can be trained with biomedical text, literature, electronic health records data, or a combination of these. The systems can also be designed for specific entity types, for example, for only gene annotations or chemical annotations, or can be general purpose for all biomedical entities. Named entity recognition tools can also be developed with different methods. So there are rule-based systems, there are dictionary-based lookup systems, machine learning or deep learning-based systems, or hybrid approaches that combine these methods together. These are examples of some tools that you may or may not be familiar with for both entity-specific and general-purpose named entity recognition. And as an example of concept annotation, this is an abstract from a paper on concept embeddings that used pub data to annotate the disease, gene, organism, and chemicals in this particular text. When we talk about ontology concept annotation, what we want to do is map these entities specifically to biomedical ontologies that we're looking for. So in this case, the Bio-Ontology, the disease ontology, or the protein ontology. Now, how does this fit into knowledge graphs? A brief background, biomedical knowledge graphs are graph structures where biomedical entities are represented as nodes in the graph and relationships between the entities represent edges. In this simplified example, entities such as phenotypes, genes, and chemicals are related to each other with different relationships. Knowledge graphs can contain heterogeneous data sources, so they can include biomedical ontologies, data from different databases, literature sources, or all of these together. They can also have existing curated information or new information, and construction and application of these large-scale biomedical knowledge graphs has become very popular for drug repurposing applications, drug target prediction, protein-protein interaction prediction, and you might have seen a lot more of them. So typically, when we are constructing large-scale biomedical knowledge graphs, we want to include knowledge that is rooted in facts in the biomedical domain. And this knowledge, more often than not, comes from biomedical ontologies such as the gene ontology or environmental ontology. And the ontologies thus serve as a backbone to the biomedical knowledge graph that we're working with. In this example, KG microbe, which was developed by the Lawrence Berkeley National Lab, contained the gene ontology, the Bio-Ontology, and CPI taxon, and the environmental ontology, in addition to other data sources. With ontologies as the backbone of these knowledge graphs, a large number of knowledge graphs further rely on unstructured data through either publicly available texts or other data sets to create nodes and edges in the knowledge graph. And this unstructured data through be it tables or literature provides additional knowledge in the knowledge graph that may not be present in existing curated data sources. So here we are referring to unstructured data as any data that has not been already mapped to ontologies or data sources that might be present in the knowledge graph. And for instance, if we wanted to include the previous abstract that we looked at into our knowledge graph, we would use either human extraction of the relations or automated relation extraction systems to extract data from the abstract, and then use named entity recognition tools to map the specific terms to ontologies that might exist in your knowledge graph. So again, with the example, breast tumor in this case, if that is the structure of our knowledge graph, would be mapped to the human phenotype ontology, the estrogen receptor to the protein or gene ontology, the NAE inhibitor to the Bio ontology, and so on. This ontology concept annotation is a crucial step when you're including unstructured data in these knowledge graphs, and it is often performed as one of the intermediate steps in construction of the knowledge graph using the named entity recognition tools. And we have several examples of large-scale knowledge graphs that have included literature or tabular data or unstructured data into their graphs. As these free texts are mapped and then including in the knowledge graph, our research hypothesis for this evaluation was that accurately mapping entities in a knowledge graph using the named entity recognition tools influences the entailed inferences, and also that it influences downstream knowledge graph applications such as link prediction. And with this hypothesis in mind, our goal was to evaluate the effects of concept annotation or named entity recognition on large-scale biomedical knowledge graphs. A good question at this point is, why do we need this evaluation? Named entity recognition tools are already evaluated for precision and recall metrics for different tasks, and this is just extra work in evaluating knowledge graphs as well. However, named entity recognition tools are not perfect. There have been studies where low precision in these tools has been shown to impact bias in word or concept embeddings, and low recall has been shown to reduce coverage in embeddings and other models as well. Disambiguation of concepts is also challenging, particularly in the biomedical domain. So we have a lot of examples where a certain term can mean many different things. And methods to evaluate these effects specifically for knowledge graphs are not readily available. So for our evaluation, we had certain requirements to choose which systems we wanted to look at. The 4 requirements that we really explicitly stated were: 1, that we should have concept annotation for free text. So an entity such as glycerol in text should be mapped to an ontology term. There should be direct mapping to biomedical ontologies. So the term glycerol should be mapped to the Bio-Ontology term here. 3rd was that we should be able to provide custom ontologies or terms as input to the system. So if Bio-Ontology is provided as an input to the system, the term glycerol will be mapped to the Bio-Ontology term. But if we're providing a different ontology, we should also be able to map that term as well. And then lastly, the one system fits all entities. So there are a lot of really good tools out there for gene-specific or chemical-specific annotations. But for this evaluation, we wanted to look at all biomedical entities mapped from one tool. Okay. So for our first use case, we looked at tabular data that was included in KG Microbe. This is a knowledge graph from microbiology, biomedicine, and the environment. It uses reference ontologies to link published data with standardized entities and relations. And it also used a named entity recognition system to map concepts from a table with free text to ontologies in the knowledge graph. So this table with substrates and pathways was included into the knowledge graph as well, using this named entity recognition tool called OntoRunner, which is a wrapper for the Ogre++ tool. And it is a dictionary-based annotator with a neural network-based disambiguation component that allows us to have custom term lists and maps free text to whichever term lists are provided to the tool. So in the case of KG Microbe, substrates were mapped to the Bio-Ontology and pathways were mapped to the gene ontology. And after mapping entities in the table with OntoRunner, we designed knowledge graph dropout experiments based on these named entity recognition results. We also generated graph embeddings using the HOPE algorithm, Disney plots to visualize what the structure of the knowledge graph would look like, and also cosine similarity distributions for positive and negative edges. The dropout experiments were included such that certain relations that were included from the named entity recognition results were dropped from the full knowledge graph to see how it would affect the structure and also the distributions in the resulting knowledge graphs. So as a high-level overview, the cosine similarity distributions here showed distribution changes that we think will impact the similarity searches as well as the embedding applications. We are working on methods and metrics to summarize the knowledge graph and the distributions that we can see here. So in the cosine similarity charts here, the x-axis is the cosine similarity values, and then the y-axis is the log of the number of edges in each of the knowledge graphs that were done with the dropout experiments. And the Disney plots are great for visualization, but not so much for interpretation, because you want to see what changes the structure really has based on which relations or which named entity recognition tools were used. And the KG micro team also has linked prediction tasks in the works. So for example, classifiers based on the knowledge graph embeddings can be used to predict the cell shape annotations for the taxa with fairly high accuracy. And in general, we can observe degradation of prediction performance by applying the knowledge graph relation dropout experiments to link the prediction problems. This use case has a more high-level evaluation of looking at the changes in structure and the prediction performance. So in our next use case, we wanted to look at more granular evaluations of how these named entity recognition tools are working and being incorporated in the graph. So for the second use case, this is NPKG, which includes a literature-based graph component from full text of scientific literature, where nodes and edges are extracted from these full texts. And there are more details of the knowledge graph construction, but for this evaluation, we focused on this step of processing and mapping the terms found in the literature to biomedical ontologies. And we used 13 open biological and biomedical ontologies for these mappings. Specifically, we use the NCBio bioportal annotator tool. It also allows custom ontologies as inputs, and it's a publicly available REST API that can be used. And for all terms extracted from these 716 articles, we use the bioportal annotator to map nodes to the 13 OVO foundry ontologies. And so what we found is we had 34% exact matches, where exact match is anything from the text that was mapped exactly to a label of the ontology term. And we had 65% non-exact matches that we were very interested in seeing what those results were and what we could understand about the concept annotation from those non-exact matches. So we looked at different entity types for the non-exact matches, including genes, chemicals, proteins, diseases, organs, body parts, and the cells. And then we also applied filters on these non-exact matches, such as the semantic type matching, the longest matching concept, extraction, removing generic annotations, and also including preferred ontologies, such as ChEBI for chemical types. And these are examples of these 9 fatty acid related concepts that were actually mapped to that fatty acid concept as a non-exact match. And then a couple of incorrect matches as well that we later manually reviewed. So after the filtering step, all of the matches were manually reviewed to see what the drop in number of concepts matched would be and how this would affect the knowledge graph later on. And so looking at the distribution after filtering and manual review, there was a definite drop in the number of concepts that were matched. Assuming that all of these concepts would be later included as nodes in the knowledge graph, this gives you an idea of how much information loss there might be if we're using just the manually reviewed ones, just the filtered ones, or all non-exact matches that we are not sure what the quality is like. And so the options after review were we could include all matches into the graph. It would have a lot of data, but it might lead to a noisy graph with non-specific inferences. We could use only the manually reviewed ones. Again, it might lead to loss of information because there are certain nodes that would just never make it into the knowledge graph. There's also an option of including everything that was not mapped at all, which certain studies have done just to not have that loss of information. And the result was that there's no perfect answer for how this would work. So we need a balanced approach without a lot of manual review. That would be our goal. And more importantly, we need to assess the impact of the named entity recognition system results on the different graph applications that we're using. We're still working on what that balanced approach would look like, but I hope that these examples of both a high-level overview and a more granular evaluation has helped make a case for why evaluation of these named entity recognition systems, specifically for knowledge graph construction, is important. I'd also like to point out certain limitations in the evaluation. We all know text is noisy when we're trying to map it, and we assumed that every entity would have an annotation, but it might not necessarily have that. We also had lenient guidelines for filtering and review when we were manually reviewing entities, so the results for the correct matches might be overinflated because correct does not necessarily mean useful when you're drawing inferences later on. And we also did not consider certain entity-specific tools and systems that did not map to ontologies that are also popular in these applications. In conclusion, I would like to say that high-throughput knowledge graph construction has gained acceptance. However, it is difficult to know and understand the contents and structure of the large knowledge graphs. Through the named entity recognition methods, we can incorporate latest knowledge from free text or structured non-standardized data before it has been formally distributed into the curated databases. And that the granular evaluation of these named entity recognition results can help expose biases, issues, and pitfalls that might occur later on with different application-based methods. We also need best practices for the evaluation. So named entity recognition, or NLP in general, has intrinsic and extrinsic evaluations for different tasks, which don't currently exist for knowledge graphs. And we are working on both high-level and low-level evaluations with different knowledge graphs as well as different NER systems to assess the impact. I would like to thank all the collaborators on this project, Martine, Harry, and Harshad from the Lawrence Berkeley National Labs, Bill Baumgartner from University of Colorado, Tiffany Callahan from Columbia University, and Richard Boyce from the University of Pittsburgh. Thank you so much for listening. If you have any questions or if you work in this space and have ideas, I would be happy to hear them.
Any questions in the room?
Yeah.
I'm wondering if you might be willing to go back to the Disney plot.
Yes.
And if you, so I think this part's really interesting, and I wonder if you might speak a bit more in how you interpret this.
That's a good point. So we have a lot more Disney plots for different nodes and edges. These ones were looking at components of the graph, and this was just for KG microbes. So all the singletons, I think, are highlighted in the yellows. We had an idea of how changing the orientation might give us a better idea of how the structure changes, but then we find out that you're not really supposed to change them that way. So cosine similarity distributions, I would say, are a better picture at this point of looking at it. These were also constructed from only one embedding method, which is like the higher order proximity preserving one. And when we tried it with a different method, it also gives different structural interpretations, or not interpretations, just visualization of it. So, yeah.
Thank you.
Perfect talk. So we mentioned that PubTer does not map to ontologies, and the author of that, I think it maps to control vocabulary. So it maps for diseases, maps to mesh and chemicals as that as well.
Yes.
So that you could be using the cross links between mesh and CABI for that purpose.
I will be honest with you, we had multiple debates about PubData because we wanted to include it since it is used so much in different named entity recognition systems. It was only when looking at the specific mapping in the indirect mappings that we would have to do that we dropped it for now. I know that it is still on the list of tools, including SciSpacey and Gilda, that we would want to assess in the future. And I did look at the mesh to CABI links, but then it was some of the other ones that I think the mesh to CABI was okay, and the NCBI taxon was okay as well. And then the disease ontology one tripped us up a little bit, and then the gene ontology also was okay, I think. And there's more components here that we wanted to include in, so sequence ontology and cell ontology.
Yeah, thanks for considering, and happy to talk more.
Yeah, that would be great.
We have one question from the virtual group. So sorry if I missed it, but how do you choose the ontologies you use for annotation? And this comes from Nurtama Bennett.
The ontologies that we use for annotation mostly came from the ontologies already existing in the knowledge graphs. So for KG microbe, for instance, we already had a constructed knowledge graph with gene ontology, CABI, NCBI taxon, and environmental ontology. For this, the choice of ontologies was limited to these. For the second knowledge graph, it's actually an extended version of the phenolator knowledge graph, which already uses those 13 OBO ontologies, which is how the choice for those was defined as well.